Yes, thank you, Sandeep. Welcome again, everyone, to come to join this special session on NADP signaling. We will start the session with the talk given by Professor Antony Galione and Dr. Margarida Ruas from the University of Oxford. Professor Antony Galione joined the Department of Pharmacology of University of Oxford in 1991. He was the chairman of the department between 2006 and 2015. Recently, he has been elected as a fellow of the Royal Society. Congratulations on that. The primary work in Antony's lab has been concerned with the mechanisms and the roles of CADPR and NADP as intracellular messengers regulating calcium signaling. Dr. Margarida Ruas completed her PhD from the ICRF in London on the role of the CDK inhibitor P16 as a tumor suppressor protein. Gita then progressed her work in cancer research at the Gooden Institute in Cambridge. In 2002, Gita joined Antony's lab, where she started and continues her research on the molecular mechanisms of NADP signaling. Antony and Gita today will be giving a talk on progress in understanding the molecular mechanisms of NADP-evoked calcium release. I think you can start the talk now.
Okay, thank you. I'll just share this.
Okay, thank you very much. Okay, I'll kick off. First of all, I'd like to thank the organisers, Sandeep and the ECS, for putting on this ultra-focused short meeting. And I'm going to give a 5-minute overview before handing over to Gita. So you could have the first slide, please. So the study of NADP-regulated calcium release has led to paradigm shifts in our understanding of calcium signaling and calcium homeostasis. But it also provides fascinating pharmacological problems in terms of its receptor or binding site, in its discrimination between two virtually identical molecules, NADP and the more familiar coenzyme NADP. And in the slide, I show the broad canonical signaling pathway for NADP. It mobilizes calcium from a non-ER store, and we later showed that it was acidic stores such as lysosomes that were the target. Pharmacologically, NADP-evoked calcium release is different from known ER calcium release channels in that it is selectively blocked by various inhibitors of the voltage-gated cation channel superfamily. This led us to converge on two pore channels, working with Mike Zhu and colleagues, as the principal pathway for calcium release activated by NADP. This, as Sandeep has mentioned, is a novel family of endolysosomal targeted channels with homologies to voltage-gated channels. And this finding has been instrumental in changing our view of the iron physiology of lysosomes beyond pH. And NADP can regulate the physiology of these organelles and cause discrete local calcium signals. In another new concept in the slide, this trigger calcium can often be amplified by calcium-induced calcium release from the ER to globalize calcium signals, probably at discrete membrane contact sites. So most of the calcium you see triggered by NADP is from the ER, with only this small little pacemaker step being the true calcium release from the endolysosomal system. However, somewhat to our surprise, NADP does not directly bind to TPC subunits in its functional homodimer. Each subunit is around 100 kilodaltons. But it's been proposed that there are separate, smaller, auxiliary NADP binding proteins, which must associate with the calcium release channel to confer sensitivity to NADP. Now, the doyen or godfather of NADP binding proteins is here, Tim Walzeth, who's been instrumental in developing this field, in helping groups around the world working on this long-standing problem. So thanks, Tim. Next slide, please. So one of the unique and peculiar features of NADP-evoked calcium release compared to other messengers are its concentration response relationships and profound desensitization by NADP. This differs between the sea urchin egg system on the left, where NADP was first identified as a calcium mobilizing molecule by Hong-Chung Lee, and on the right, mammalian cells. In urchin, sub-threshold concentrations of NADP can fully desensitize release by normally effective concentrations, and we call this type 1 desensitization. However, the situation is reversed in mammalian cells. Here we found in pancreatic acinus cells that higher concentrations of NADP actually inhibit calcium release in this mechanism. And this is reflected in the now famous characteristic bell-shaped curve. And this is type 2 desensitization. And this may be correlated with radio-labeled NADP binding studies between these systems. A high affinity single binding site in the sea urchin membrane, whilst there are two populations of NADP binding sites in the mammalian cells, a high affinity and low affinity. So this may indicate we need to look for multiple binding proteins, and there may be a difference between the classical urchin egg system and mammalian cells. So my final slide, please. So how might these NADP binding proteins work? I show here some possible scenarios for activation. And for pictorial simplicity, I've not distinguished between NADP binding proteins that are soluble, which may be mainly the case in the mammalian systems, and those that are integral membrane proteins, as in the urchin. So if we look at this first scenario, the auxiliary NADP binding protein may be pre-bound to the TPC channel, and when NADP binds it, it causes a conformational change to open the channel. The second possibility is that NADP binds to a protein which is separate from the TPC, and then the activated complex is then recruited to the channel to activate it. Thirdly, the binding protein is pre-bound and inhibitory to the channel, and NADP binds to it and causes it to dissociate, thus disinhibiting the channel. And fourthly, the NADP binding protein complex may be, for example, an enzyme which is activated by NADP, and this could post-translationally modify the channel, leading to gating. So in summary then, NADP binding proteins must satisfy 4 criteria. They must bind NADP with high affinity. Secondly, they must discriminate between the very low concentrations of NADP and high concentrations of NADP in the cell. Thirdly, knockout or knockdown of the protein must block NADP-induced calcium release. And fourthly, they must dissociate with the target channel, such as TPCs. So Guido Ruas has been working on the molecular mechanisms of NADP-evoked calcium release for almost 20 years. I think you all would agree it's a tough problem. She, for example, created TPC knockout mice, which were instrumental in demonstrating their key role for NADP-mediated calcium release. And she'll outline how she cloned the sea urchin egg TPCs and showed that they were associated with high affinity NADP binding. She's now going to give us an overview of some of our work and the strategies we've taken in studying NADP binding proteins. We're still in the process of characterizing various candidates because of our demanding comparative approach between urchin and mammalian systems. But I gather later we'll hear about at least 3 candidate proteins this afternoon. It's a bit like London buses. You wait for 20 years and then 3 or more turn up at once. So Guido.
Okay, thank you, Antony. Okay, so to remind us ourselves about the characteristics of NADP binding in sea urchin, as Antony already mentioned, we see a very high affinity binding site in the sea urchin. It's a single population of binding sites, and it discriminates really well with NADP. Another characteristic, it seems that this protein is completely membrane bound, hardly anything on the cytosol, and the only kind of treatments that release it for membrane bound fraction to a soluble fraction is by treatment with detergents. So none of the other treatments that I've tried can do this. One really interesting feature that we see in NADP binding in sea urchin is the apparent irreversibility of binding in the presence of potassium ions. So you would expect that if you add cold NADP to a mixture, pre-bound mixture of hot NADP with its binding protein, you would expect it to be able to displace it and dissociate it. And in fact, you can do this, but only in buffers that are free of potassium ions. So this is a very cool feature that can discriminate binding proteins. Another property that we know is that this NADP binding in sea urchin interacts with TPCs, and this is at the endogenous levels, so it doesn't require overexpression at all. And we were able to show this through the development of antibodies against the 3 isoforms that are present in sea urchin, TPC1, 2, and 3. And antibodies that we've developed against TPC1 and 3 were able to immunoprecipitate binding activity from solubilized homogenates, and with properties that really mirror the ones seen in the native membranes. So very high affinity binding for NAADP and discrimination with NADP, and also the inability of dissociation in the presence of potassium. So if you remove potassium, you can wash off the pre-bound NADP, but not in the presence of potassium. Some years ago, it was George Berridge tried to ask the question, how big were these NADP binding proteins in sea urchin? And by doing 2 different types of separation, they found that the NADP binding protein were present in very high molecular weight complexes, that the sizes were somehow different depending on the technique that they've used. But a really breakthrough was the development of the photoaffinity probes developed by Tim Walseth that were able to be covalently linked to its binding partner by shining UV light. And because we can also radio-label these probes, then you can run the membrane proteins or even homogeneous proteins that have been labeled on SDS page, the naturing page, so that you can reveal the real sizes of these labeled proteins. So doing that, it's a very obvious labeling of 3 protein bands. And by doing competition with NADP, we can see that the affinity is very similar to the one that we obtained doing radioligand binding assays. And again, this binding is very specific for NADP. It is really important to see that these proteins also show the potassium-dependent irreversibility of binding, and they are also present in the TPC1 and the TPC3 IPs. Okay, so for a long time, we tried to purify these NADP binding proteins from solubilized sea urchin membranes and purify them through chromatography procedures and also using NADP affinity columns, but they were not successful. So we thought that maybe we could go back to our TPC3 IPs and see what proteins were pulled out that could be potential NADP binding proteins. And the reason why we thought this approach would be worth considering is because we had 2 antibodies that we have developed against TPC3. They come from 2 different rabbits immunized with the same 2 peptides corresponding to TPC3 sequences. And we could show that both of them were able to IP the sea urchin TPC3 protein. But if you did radioligand binding assays in those immunoprecipitates, only one of them could recover the NADP binding protein. So we thought that maybe if we compare the proteins that are present in this IP versus this one, we could have a look at potential candidates. So we started by cleaning up these peptides and affinity purifying them against the antibodies and affinity purifying them against each of the peptides. And we could see again that only one of these antibodies were able to bring down NADP binding proteins. And just to make sure that we hadn't to destroy the capacity of these 3 antibodies to bring down proteins, we've performed STS page and Kubasi labeled the proteins. And we could see that the antibodies that have been purified against peptide Bs were really able to bring down proteins at similar levels, but only this one brought down the NADP binding protein. So by doing mass spec, using these antibodies in triplicate, we can see that the CBIP is our negative control and the FBIP is our positive samples. And after doing a protein identification and label-free quantification, we could kind of plot the enrichment fold of each single protein that we've identified. So this is enriched on the FB over FCB. And deciding on an enrichment fold, a threshold of enrichment fold of 3, we've zoomed on 61 proteins that would be worth studying. Now, you probably have already spotted this one here, right at the top. So each spot corresponds to a protein that we've identified. And this protein here is a solid carrier protein. It's a very high molecular weight, much larger than what we'd expected from the photoaffinity labelings. But one interesting thing is that one of the protein interactors that is actually a regulator of this slick protein in mammalian cells was also pulled down in this IP. And it is a molecular weight that is similar to the ones that we've observed by photoaffinity labeling. So in the process of analyzing these proteins, and I would like to actually come back to this one a bit later on. Okay, now moving on to the mammalian binding proteins, the situation of the features of NADP binding in mammalian tissues are very different from the sea urchin. So as Antony has already mentioned, you see two populations of binding site, one with a high affinity in the low nanomolar range and another one lower affinity in the low micromolar range. And another thing that we actually see is that although we do see binding on the membranes, there's a huge amount of binding also on the cytosol. And in fact, I think these two population of binding sites are actually more easily visible in the cytosolic samples. And again, the high affinity binding site is discriminatory between NADP and NADP, but not the low affinity binding site, and also not touched by NAAD. Okay, so in mammalian samples, we also see interaction with TPC2. So this was done either in membranes from cells overexpressing TPC2 that we recover more binding and compared to wild-type cells, but also via TPCs against, IPs against TPC2. And what really kind of cemented the idea that the binding that we were seeing was due to an accessory protein was studies using tissues from double knockout mice that we've developed and shown to be a knockout for both TPC1 and TPC2. So if we compare the pattern or the characteristics of binding in tissues from wild-type animals and TPC double knockouts, either by radioligand binding assays or by photoaffinity labeling, we don't really see many differences between the two samples. If in fact actually more binding maybe than wild-type, but we could discuss this further. But the two populations of binding sites are still present. But by photoaffinity labeling, we could see that there was really almost just a single protein whose labeling was computed by co-incubation with NADP. So it shows that there was a very highly specific protein in both animal tissues that was binding NADP. So we decided to try to purify these proteins using a completely unbiased approach. So it doesn't rely on the ability to bind TPCs or any other channels. It just relies on separation of proteins and following the presence of NADP binding proteins using a radioligand binding assay. And I could show that by doing separation using a hydrophobic interaction chromatography, I could separate the high affinity binding site from the low affinity binding site. And this is probably due to different proteins. Another method that I can see can separate the high affinity binding population from the low is by doing gel filtration, which separates proteins according to size. And I could show that the high affinity binding population corresponds to a molecular size between 20 and 30 kilodaltons. So you see a pattern emerging here. So what we decided to do next was to sequentially purify this protein using different chromatographic procedures. and check the degree of enrichment in each one of the steps. So after gel filtration, after ion exchange, hydrophobic interaction, and gel filtration, I could see a very high enrichment of this NADP binding protein by radioligand binding assays. And also by photoaffinity labeling, you could see that this protein was also enriched. Doing mass spec analysis on these proteins that were obtained in the last elution from the gel filtration, and then doing a filter by size and looking at the shared candidates between the 3 replicate experiments, we went on to do virtual screening with NADP docking to see if we could see what proteins were the most likely to interact with NADP, and we selected 3 proteins to do binding assays. One of these proteins could show complete discrimination between NADP and NADP. However, the KD was in the low micromolar range rather than in the low nanomolar range, and we could discuss this maybe later on. Okay. There is another technique that we've employed recently to study TPC1 and TPC2 interactomes with a view to potentially identify new interacting proteins. So this bioID technique relies on the proximity biotinylation of interacting proteins due to adduction of a promiscuous biotin ligase that we tag to our protein of interest, in this case TPC1 and TPC2. And this labeling is done in live cells in situ, so protein interactions that we recover can be quite physiologically relevant. And also in comparison to other methods such as affinity purification, it has a potential to better identify transit interactions or weak interactions or even low abundance interactors, because it integrates events that occur during a period of 24 hours. Also, after labeling, we solubilize the cells with stringent conditions, including buffers that have SDS, and this will minimize the recovery of false positives due to potential membrane carryover. So we've done this analysis and identified proteins by mass spec and quantified them by label-free quantification. And we were able to assign a TPC1 interactome and TPC2 interactome by selecting proteins that were enriched in the TPC1 samples over control, and the same for TPC2. And then we could do like a Venn diagram with proteins that had already been described as TPC1 and TPC2 interactors by affinity purification, for instance, IPs. And we could see that there were a lot of proteins, potential novel interactors, and also a small proportion of them that had already been described as TPC1 or TPC2 interactors, and they are shown here on the right. Okay, another type of example of analysis that we can do from this is by directly comparing proteins in the TPC1 and the TPC2 interactomes, and see if they are preferentially enriched in one over the other. So as a very nice control, you can see that TPC2 is the most abundant protein in the TPC2 interactome, and TPC1 again in the TPC1 interactome, because bioID has also labeled this protein, these 2 proteins. And also in the middle are the ones that really don't show much discrimination between the 2 TPCs. So this will be kind of a really good resource to look at potential candidates that might be regulators of TPC, but also effectors of TPC functions, so essentially proteins that are downstream of TPC. And by looking at these proteins, we can define novel pathways where TPC1 and TPC2 might be involved. So we've started characterizing some of these proteins in these interactomes, and we've been concentrating on common proteins that we've detected in the sea urchin TPC3 IP. And one of them is the protein that I have mentioned that is a regulator of the sleek protein that I've mentioned before. So I've confirmed the interaction using co-IPs, and you see that when I express that interactor together with TPCs, and then do an IP via the candidate interactor protein, I can pull down TPC1, I can pull down TPC2, I can also pull down TPC2 that is targeted to the plasma membrane, but I cannot pull down another lysosomal protein such as LAMP1. So this shows that the interaction is specific. It's not just that I'm bringing any kind of protein in the lysosome. So the validation of some of the interactions are on the way by doing co-IPs and also NADP binding proteins, by doing radioligand binding proteins of these candidates. And today I won't be able to talk more about this, but this is the type of assays that we are carrying on, mainly now from the TPC2 interactomes and the Urchin TPC3 IPs. So thank you, and if you have any other questions, feel free to ask.
Yeah. Thank you for the talk. And I'm afraid we can only ask 1 or 2 questions given the time we have. So if you have questions, could you please raise your hand or just unmute and ask.
The raise hand button is at the bottom.
Yeah, it's at the bottom of the screen.
I don't see anybody.
Yeah. Any questions? You can really see them, see everyone. Not really see anyone. Okay.
Okay, Sandip has raised.
Okay, Sandip has raised. Sandip?
Yeah. I have a question. So Gita, this regulator that you talked about, so that you found that in the mammalian samples and you found it in the sea urchin samples?
Yeah.
Okay, so that's nice to see that across both, right?
Yeah, so I've detected it on the TPC3 IP in the sea urchin, and it was also pulled down in the TPC1 and in the TPC2 interactome.
And what's the location? And presumably you can't talk too much about it, but is it consistent with it being in
So the sleek, that one that was very, very abundant in the TPC3 IP in the sea urchin, is a membrane protein. But the interactor has also been shown to interact with vesicles. They don't say what is the partner. We don't know what is the partner. But if you express it, you can also see some localization in vesicles. They haven't really defined which type of vesicles. And I've confirmed that I can actually see internal localization. But it's very, very abundant at the plasma membrane.
Okay. Interesting. Thank you.
Yeah, and we have one question from Professor Christian Scream. So the question is, in your pull-down assays, did you check for tryptophan or ryanodine receptors?
Sorry, say that again.
Oh, in your pull-down,
In your pull-down assay.
yeah, in your pull-down assays, did you check for tryptophan or ryanodine receptors?
They don't come down. So in terms, so the sea urchin TPC3 or the interact on the TPC1 and 2 interactome.
Yeah, I think
So on the pull down.
Yeah, Christian is here.
Sorry, Gita. The question is, I think, did you? So you tested for TPC1 and TPC2, and you could pull down both, right? But did you also do an experiment with TRPML chance or ryanodine?
Oh, I see. No. So I haven't done that yet. No. Yeah, you're right. Yeah.
Right. Thanks.
Okay, I think we need to move on.